Thank you, Josh, for the introduction. And I'm sorry that the slides were made in PDF and it's not so easy to do the switching just now. So it's my great pleasure to present our simulator sCDesign3, as I briefly mentioned in my morning talk. And so this talk will be a little technical because I'll talk about the details about how we did the simulation. And this is my student Dongyuan Song's work, but unfortunately he cannot be here because of visa. So I'm presenting on his behalf. So first, our goal here is to generate count data as in the process data for single cell and spatial omics. So they will consist of cell by feature matrix and a cell covariate matrix. So specifically, the cell by feature matrix can be gene expression, as in single cell RNA-seq and spatial transcriptomics. The features can be peaks, as in the chromatin accessibility data, such as single cell ATAC-seq and snare-seq, and it can be proteins, as in protein abundance in site-seq data. So these are what we call features. And for cell covariates, we consider them in two categories. The first set of covariates are for cell heterogeneity, including discrete cell types. This could be considered as known if the cells are annotated or latent if you do clustering. And it could be continuous trajectories like pseudotime, and these are usually latent and inferred from data. Or they can be spatial locations, and these are known for spatial data. So this is the first set of covariates. The second set of covariates is regarding experimental design. It can include batches, and the effects of batches are unwanted, and they could be conditioned, and here they are biological signals. So the reason we developed this sCDesign, which is the version 3 right now, is for two purposes. I should say three purposes now. The first one is, of course, computational benchmarking. And so there are a lot of computational tools for single-cell RNA-seq data. And for users, how to choose those tools is challenging. And also for method developers, how to do benchmarking is challenging as well. Second, we want to do model inference. That means we want to learn from real data besides simulating data. Our argument here is that if our model can generate realistic data, then it means that the model is reasonable. It can fit well to real data. Then we can use the estimated model parameters from real data for interpretation. So this means that what we can do is that conditional on a cell covariate, could it be cell type, pseudotime, or spatial location, we can infer every gene's conditional distribution and every gene pair's conditional correlation for biological interpretation. And besides, which I think is most exciting, is related to my talk this morning in silico control experiment. Can we generate negative control as synthetic null or positive control to evaluate a pipeline of methods, false discovery, and discovery power? So all these causes caused us to develop a realistic simulator with interpretable parameters. So there are a few challenges in single-cell spatial omics data, multi-omics data, including the high dimensionality of features. It could be larger than the cell number, and the correlation structure of features, and the diverse covariates, cell types, continuous trajectories, spatial space, and the multi-omics, like the RNA part and ataxic part and protein part, they may have different distributions. And also, we hope the model parameters can be transparent. So this is the published paper, sCDesign3. And so these are the two main illustrations of functions in sCDesign3. The left one shows the simulation functionalities, which we use to verify that our model assumption is reasonable when we have well-annotated cell types, trajectories, or spatial locations. So in this case, we want to show that our synthetic data mimic real data very well. And for multi-omics, these are the features we can consider to generate. And for experimental designs like the biological covariates, gender, age, disease health, or the batch effects can be generated. The right part is about interpreting real data, which means that using the model parameters, we can check whether a gene has the same mean or different means between two cell types, how a gene's mean expression change along a trajectory, or how a gene's mean expression change in the two-dimensional space. And the model selection part, I think, is very interesting because our model is a probabilistic model, so it can give us a likelihood. Based on that, we can compute the Bayesian information criterion, or BIC, that can evaluate how well does our model fit to the data given different trajectories. Say we have two different trajectory inference methods, we take each inference result as the covariance, cell covariance into our model. Then we can compute the BIC. What BIC measures is the goodness of fit of the model to the data. So a lower a BIC, the better the model fitting. So we can say, yeah, this trajectory fits data better. And for clustering, we can do the same thing. Which set of clusters by Surratt or another method fits the data better? Finally, for the model alteration, this is what I talked about in the morning. Given what we fitted on real data, we can modify the model parameters to generate the corresponding synthetic data. Like the real data has two cell types, we can generate cells of one cluster, or we can make a gene as not differentially expressed. So specifically, what's the method? So I used the following notations. This is a technical part. I denote Yij as the measurement of feature J in cell I. Feature J could be a gene, and this Yij is usually a count. The second matrix is the cell by state covariance matrix. So here, this is the first set of covariates we're interested in. They can be cell types, or if it's cell type, then we have just one dimension because the cell type is a categorical variable, or it could be a cell pseudotype in P lineages. That means we will have P pseudotypes per cell, or it could be the two-dimensional spatial coordinates per cell. So we're interested in this covariant, how it's related to gene expression. And then another set of covariates is a cell by design covariance matrix. So for example, here we can consider a batch effect, batch vector, and a batch indicator and a condition indicator. So again, for every cell, we have a batch and we have a condition. So first, in our model, we consider every feature's marginal distribution first. So for every feature J, we fit a model in the most general form is as follows. We can use this method developed in statistics back in 2008 called Generalized Additive Model for Location, Scale, and Shape, short as GAMLSS, GAMLSS. So what it does is that we can assume the marginal distribution of feature J in cell I, Yij, given cell I's state covariance, design covariates, to independently follow a distribution, a count distribution that depends on cell I's two covariates, and with those three parameters in the most general case. So mean, variance, and zero proportion. So for each parameter, we model its relationship as in the generalized additive model, which is a generalization of the generalized linear model, like logistic regression, many people are familiar with. So in each relationship, we assume the following. There is an intercept, which is fixed just for every feature J. And we have a batch intercept effect and the condition intercept effect. And for the state variable effect, we consider to be possibly nonlinear if the state covariate is a trajectory, like pseudotype. So we don't assume the gene expression to be linear with the pseudotype, but we allow it to be nonlinear. And also for spatial data as well. Okay, so assuming we can fit such a model, then for feature J, we will get a fitted distribution given the cell's two sets of covariates. So these are the distributions our model can allow for: Gaussian, Bernoulli, Poisson, negative binomial, and the zero inflated Poisson and negative binomial to account for different features and their distributions. So for the cell state covariates, as I mentioned, we can consider cell types, one lineage, P lineages, and spatial location. So the joint modeling part is the Gaussian copula I briefly mentioned in the morning. So specifically, after we get a marginal distribution for every gene, for every feature J. So we have F1 to Fn. We have a total of n features. So what the copula does is that it models the joint distribution of the n features after each feature is transformed by its own CDF. So this is CDF1 to CDFm. So the idea here is that after transformation, every feature becomes uniform between 0 and 1. And copula links those n uniform 0 to 1 random variables into a joint m-dimensional distribution. So specifically Gaussian copula, what it does is that for each uniform 0 to 1 variable, it applies the inverse Gaussian CDF to turn it into a standard Gaussian random variable. Then we would have a total of m standard Gaussian random variables, and their joint distribution will be modeled by an m-dimensional Gaussian distribution with zero mean, univariance. What's left is a correlation matrix, which is equal to the covariance matrix in this case, because the variances are 1. So we are left to estimate this R. So one more thing I should say here is that to account for the high dimensionality in Gaussian copula, when the number of features is larger than the number of cells, this will give us a non-invertible correlation matrix because of the high dimensionality. So this will give us troubles with finding or calculating the likelihood. To counteract this issue, we use a technique called Vine copula, which I think is a very smart technique that decomposes a high-dimensional m by n matrix by this vine. You can consider this to be a tree. So what it does is that it decomposes this high-dimensional correlation matrix by pairwise correlations between the highly correlated features in the first layer, and then the second layer will be conditional correlations given the first layer. The third layer will be conditional correlations given the second layer. So what this can do is can give us a way to approximate the correlation matrix and make the likelihood function computable. Okay, so to estimate this copula, we use the plugin approach, which is a jargon in statistics. What it means is that we take the fitted marginal distributions for each feature j, we transform each feature j's count in cell I into this uniform 0 to 1 random variable to make this variable continuous because the counts are discrete, right? If you just do the transformation, it will still be discrete. To make it continuous uniform 0 to 1, we apply the so-called distributional transformation, which is essentially an interpolation of two discrete values by doing this random interpolation to make it continuous. Then this Uij becomes uniform 0 to 1. And then what we do is that we apply this Gaussian copula to those uniform random variables to estimate the Gaussian copula correlation matrix. So finally, with the model estimated, we have a Gaussian correlation matrix estimated and every feature's marginal distribution estimated. We do sampling to generate synthetic cells. So we first sample from this m-dimensional copula, which will give us m-dimensional uniform random variables. Each is a uniform 0 to 1 random variable. After the sampling, using the fitted marginal distribution from the previous step, we just apply the fitted CDF's inverse to each uniform random variable to convert this uniform to counts. And that's how we get a synthetic count for feature j in synthetic cell I prime. So here we use I prime to indicate that the synthetic cell doesn't correspond to any real cell. It's an independent sample from the fitted distribution, but the feature j is the same j. So to quickly go over the results, these are all from our published papers. So we can show that we are probably, I think back then, we can say for sure back then, we are the only simulator that can take trajectory as an input to generate data. So that's why we can see the colors indicating the pseudotime values in our synthetic data, while other simulators didn't directly use the pseudotime. So that's why we show that. So here in terms of this mixing, how well does the real cells mix with the synthetic cells? And this value should be at most 2 and at least 1. So closer to 2 means better. So we have better this value. And we can show that the other 3 methods, even though they can do pretty well with simulating cluster cell types, but they are not designed for simulating trajectories. And as again, I mentioned that briefly in the morning. And this is how we can do for spatial data. So here we are generating the 10x Visium data, and the top row shows real data, bottom row shows synthetic data, which means that using that generalized additive model with location, that GAM LSS, we can actually capture the mean function for every gene pretty well. And given that, we can generate counts. And this is showing how we can generate the site-seq data, which has both protein and RNA. So top row real data, bottom row synthetic data. So we can see that we can even preserve the protein RNA correlation. So they are pretty well prepared. Finally, other student Guang Ao developed this read simulator. So coupled with our scDesign3 for count simulation, it can generate sequencing reads for benchmarking low-level tools like the p-calling tools or UMI deduplication tools. And this one shows that for single-cell ATAC-seq, we can mimic the counts well, and we can also mimic the read coverage pretty well. And this paper is under revision in Nature Communications, and you can see the bioRxiv here. Finally, to show that the model parameters can be useful for biological interpretation, we're showing the correlation matrix from Gaussian copula here. So we have 4 cell types, which we're showing is that, so the 4 cell types correlation matrices are not identical. In particular, if we look at the 2 T cell subtypes, they are more similar than the other 2 correlation matrices, which means that the correlation matrix carries some biological information about cell types. And the Vine copula shows a Vine representation of each correlation matrix, so we can use it to interpret the most highly correlated genes as the first layer, and also the genes which are at the root can give us some biological interpretation. So we leave this for future biological studies, even though we haven't really done that in this paper. Finally, you have seen this from my morning talk, we can generate synthetic null, and this cannot be done if you just permute the cell type, cell cluster labels. So I want to thank my students Dongyuan and Qingyang for leading the efforts in scDesign3, and also Guang Ao for developing the RegSys simulator and the funding agencies. Thank you.
Thanks, Jessica. Very nice talk. So I have a question regarding the spatial data.
Yeah.
So spatial data always comes with an H&E image. So do you have a simulator that can generate data like conditional to the H&E image?
That's a very good question. Yeah, my answer is that it is something we are working on right now to use the H&E. But right now, our results are only based on the spatial transcriptomics data. But I do know that James Zhou has some work about predicting spatial transcriptomics data from H&E. That is not considered as a simulator. I think it's called prediction. But that's a very good question. I think finding the connections between H&E and spatial transcriptomics data should be the thing to do. Yeah.
So my question is also along similar lines.
Yeah.
So for spatial data, you looked at the gene expression correlation, right?
Yeah.
With your simulation and real data. But did you also look at the cell type proportions? For example, many of the deconvolution methods, they want to look at the cell type proportion. They want to find out the cell type proportions at each spot.
That's a very good question. So I have 2 answers. First answer is that if we just use the spatial transcriptomics data, like the spot resolution, like Visian, without the cell type information, our synthetic data will not give you cell type information. But in our paper, we have added a result in which we use the cell type proportions inferred from another method, say CyberSort-X, which is not designed for spatial data. They can do it per spot. We take that as the ground truth and we do the simulation and to show that, okay, once we use the synthetic data, pretending the ground truth is reasonable, how well do the methods like the ARC-CTD card perform on the deconvolution? So we do have that in our paper.
Okay.
Thank you.
Jessica, thank you. My question is quick. It's about perturbation.
Yeah.
And I was wondering if you had thought to use this framework to study perturbations, because the spatial will give you the constraints, but some perturbation cannot be biological real. But at the same time, I was wondering, how can we simulate data so that we have this negative control when we perturb data for cells, for example, with drugs, or simply by remodulating, for example, working in oncology, remodulating the tumor microenvironment. So what is the biological noise and what is the real perturbation? We don't have a negative control for that.
That's a very good question. I would like to talk to you more about that because I feel like to generate some negative control that's reasonable, we need biological knowledge. So but hopefully our simulator can account for that into the every gene's mean function. So we can specify the mean function in a way that there couldn't be a signal, for example. Yeah, thank you.